principles of Agrose gel electrophoresis. Made by Grace Fay and Hannah Leahy. What are the uses of gel electrophoresis? Well, there are many. These uses include testing DNA for genetic diseases, paternity tests, and testing evolutionary relations in organisms. A beneficial use of gel electrophoresis is testing genes for a particular disease. The steps involved include separating the DNA from white cells and storing it, purifying the DNA, and producing millions of copies of it using PCO reactions, which are polymerase chain reactions which amplify a single or a few copies of DNA, generating thousands to millions of copies of a particular DNA sequence. The DNA is then cut into fragments using specific enzymes. Where the DNA is cut and the size of the fragments is unique to each person, the DNA fragments are then separated using gel electrophoresis. If the fragments are only found in people with the disease, this suggests the fragments contains DNA from a gene variant. This suggests the person is more susceptible to the disease. The method for preparing the Agrose gel molds includes weighing out the Agrose and then adding the TBE buffer to a large mixing bowl. It is then heated to dissolve the Agrose and cooled down to about 60 degrees Celsius. Ethidium bromide or gel red is added and mixed in. The solution is poured slowly into a mold to avoid bubbles. The comb is then inserted and it is allowed to set for about 30 minutes. TBE buffer is added into the mold to cover the gel to about 2 to 5 millimeters of buffer.
This is a drawing of the horizontal limit of Agrose gel. The wells should be placed at the side with the cathode. The sample will run from the negative to the positive as DNA is negatively charged. Running buffer is added until the level is about 2 millimeters over the level of the gel. The running buffer keeps the pH at neutral. The DNA samples with the loading buffer are added to the wells. The loading buffer allows the DNA to sink to the bottom of the cell. The samples are added using a pipette. The pipette should be held vertically over the unit, making sure to keep the pipette in the buffer at the opening of the well, but avoid damaging the well. Set the current to run through the unit, and as the time moves on, one will notice that the DNA samples moving through the gel are at different rates.